Good day, everybody. My name is Vinay Pho, Senior Product Manager at Advanced Cell Diagnostics, and I'll be discussing our new product introduction of RNAscope 2.5. Also, Dr. Courtney Edison, our R&D scientist who has extensive experience in molecular and cellular biology, will be discussing a case study on RNAscope 2.5 robustness in the lab. A few points about the webinar. The audience will automatically be muted, and if you would like to ask a question, please wait till the end and select the chat button to ask questions to the hosts and presenters. Apologies in advance if we are unable to answer your questions, and we will follow up with you offline. For today's webinar, we'll start off with a brief introduction to RNAscope in general. Then we'll introduce the RNAscope product line and what has changed and what has been improved. And then Courtney will discuss the key highlights of RNAscope 2.5. And now a basic introduction to RNAscope. RNAscope is based on a proprietary in situ hybridization technology based on a unique probe design and signal amplification. This platform has a sensitivity to detect most genes in situ, as well as simultaneously quantifying multiple RNA transcripts. Just as any in situ assay, RNAscope assay involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to an RNA target of interest. The specific signal is amplified using a double Z probe system, which is subsequently detected and quantified. The assay is extremely universal as well as rapid and scalable. Other webinars for more detail on how this works and the video are located on our homepage. It's important to note that for our RNAscope 2.5 assay, nothing is changing, and that nothing is changing from the amp structure and probe design, and all of the same concepts apply. So here we present a snapshot of ACD's history, where the company was founded in 2006. Through various product launches, we have continued to innovate based on customer feedback. We launched our first version of RNAscope in 2010. Adoption of our technology has grown through the years, and now as the company is expanding, we continue to listen to our customers and have just recently launched RNAscope 2.5. Our growth and adoption is best exemplified by the number of peer-reviewed publications. In 2013, we had close to 1 publication per week. And in 2014, we saw 84 publications, or 7 per month. So far, in the first 3 months of 2015, we observed 50 new publications, so averaging 16 new publications per month. We anticipate that 2015 publications will exceed those in 2012, 2013, and 2014 combined. And here the images are examples of 8 journal covers for the 15 publications in January 2015. And now we'd like to introduce our latest product platform, RNAscope 2.5, which has just been officially released. RNAscope 2.5 covers single-plex chromogenic and pretreatment reagents for manual, Leica, and Ventana automated systems. The product highlights an improved robustness, an ease of use, and improved performance. Now to highlight some of the high-level improvements of RNAscope 2.5 versus 2.0. RNAscope 2.5 demonstrates an improved sensitivity with difficult tissues. We know that a lot of time is spent on optimization. One of the key goals for RNAscope was to provide better pretreats for better penetration, resulting in better sensitivity. More specifically, we are releasing new pretreatment reagents to be used with our 2.5 detection kits. We have a new target retrieval formulation for manual and VS, and we are also introducing a new protease plus for use with this new target retrieval. Also, we are introducing a universal pretreatment reagent bundle that combines all of the pretreatments in a particular kit. For RNAscope 2.5, we're also focused on lot-to-lot reproducibility of our detection kits. For the for the amplification components, we are now using high-quality grade ingredients with a more stringent quality control, as the assay and technology will be used in clinical programs. It's important to highlight that there are no changes to the amplification tree and structure. And lastly, with RNAscope 2.5, there is an enhanced usability with minimal hands-on time, meaning there's no need to warm the detection components. And with RNAscope, there's now a fully automated workflow for both the Ventana and Leica systems. And in many tissues, there's a reduced need to optimize pretreatment conditions. And most importantly, just to note that this is a replacement product with improved robustness at the same price. So now let's dig into more detail about RNAscope 2.5 for manual chromogenic assays for red and brown detection. First, let's start with a review of the overall workflow. For RNAscope 2.5, the overall workflow remains the same. For new users, this overall workflow is similar to IHC, but with subtle differences with tissue pretreatment and optimization. The first stage is to prepare the samples, where samples are fixed, dehydrated, embedded, and cut. For RNAscope, tissues are baked and deparaffinized. Then for pretreatment, tissue pretreatment, samples go through a blocking step to block endogenous peroxidase. Samples are then permeabilized using target retrieval reagents, and then the protease digestion is to enable better probe accessibility. Lastly, RNAscope assay workflow is initiated with the hybridization of the probe, followed by 6 amplification components, and then finished with detection of the signal. And so what's changing? What's changing in the singleplex manual workflow for RNAscope 2.5? For those that are new users, just to note, there's a variety of name changes. And for experienced users, what we're highlighting are the various pretreatment and component changes throughout the assay. So what you're familiar with, such as pretreat 1, is now being renamed to hydrogen peroxide. While this is renamed, it is the same formulation as our pretreat 1. For pretreat 2, this is being renamed as target retrieval, and this indeed is a new target retrieval formulation. And we are also highlighting a new target retrieval protocol available for use with an electric steamer. What's new with 2.5 is the introduction of Protease Plus. This is a new protease to be used with the new target retrieval, and it is a weak concentration compared to our other proteases. Some of our users may be familiar with Pretreat 3 and 4. This is Protease 3 and Protease 4. Both are the same formulations as previous. Protease 3 is of a medium concentration, and Protease 4 is a strong concentration. As I mentioned earlier, we are also introducing a universal pretreatment reagent bundle, where this contains all the pretreatment reagents, such as hydrogen peroxide, target retrieval, Protease Plus, Protease 3, and Protease 4. The probe will remain the same in RNAscope 2.5, as there's no changes to the probe design or buffer. And lastly, the amplification components 1 through 6. For our RNAscope 2.5, we are introducing new components, new amplification components, all through 1 through 6. While there are changes to the amplification components, there's also now no need to warm up these components. And lastly, as mentioned previously, there's no change to the structure. So to clarify about the reagent kit configuration, for new users, a reagent kit contains everything you need for FFPE tissues. This includes the pretreatment, detection, and wash buffers. Always use the reagent kit if you're using FFPE. So if customers decide to use other tissues, the universal pretreatment bundle also contains all the necessary reagents for different tissues. For additional information, please consult the acdbio.com website. Within the support section, our user manuals are posted based on platform type, as well as different tissue types. So after selecting a particular sample or tissue type, you can then select the particular detection kit, and all corresponding manuals will be presented. On a separate point, RNAscope Duplex and our fluorescent multiplex kits remain unchanged. These still remain as 2.0, but will ship with the new target retrieval, new pretreatment reagents such as target retrieval and protease plus. We do encourage you to refer the new pretreatment matrix on the website. So here's a presentation of the manual 2.5 assay across several targets, including a typical control, DAPB, and several tissues, highlighting the robustness of the assay. With RNAscope 2.5, there is a reduced need to optimize where most tissues will work with our standard conditions. As you can see here, we see punctate staining in a clean background. And now moving to RNAscope 2.5 LS assay. For those who are not familiar with this technology, RNAscope 2.5 is also available on the Leica Bond RX system. Here we have a specific RNAscope LS kit combined with the Bond detection kit to run on the Bond RX to yield optimal staining results with improved sensitivity and cleaner background. Similar to the manual assay, now the RNAscope assay workflow is fully automated with pretreatment staining and amplification all on the instrument. So now product details about RNAscope 2.5 LS reagent kit. As you see here, the target retrieval components remain the same. In this case, we're still using our Leica's target retrieval components. The protease remains the same. The wash buffer has been renamed to LS rinse, and this is a new formulation which is ready to use and no longer needs to be diluted or warmed. The probe component for 2.5, we are now using LS probes 2.5. These probes contain a new buffer, but have the same probe design. For the amplification components 1 through 6, we are now using the new RNAscope 2.5 amplification components. Where similar to the manual assay, there are changes to the components. There's no need to warm up these components, and lastly, there's no change to the amplification tree. The detection substrate still remains the same. And lastly, the software is new, and it is now to accommodate the new RNAscope 2.5 assay. So please contact your ACD support to schedule an RNAscope LS 2.5 training and software update. And now an introduction to the RNAscope 2.5 assay on Ventana's Discovery Ultra and XT. Similar to the RNAscope 2.5 LS, we have our 2.5 VS reagent kit for red and brown. RNAscope is fully automated on the Discovery Ultra and XT. On the Discovery Ultra, with RNAscope 2.5 on the Discovery Ultra and XT, we're able to expand the applications on the Discovery instruments. What this means is that we're able to eliminate the dependency on various containers within the instruments, and this allows us to use other chromogens for other assays on the same instruments that may also be running RNAscope. We're no longer using various bulks, such as the option wash bulk. And lastly, this results in overall less bulk reagent usage. More specifically about RNAscope 2.5 VS reagent kit, for the target retrieval, we are now using ACD's target retrieval, and this is a new formulation for better tissue permeabilization. The protease components remain the same. We are introducing a DWAX, which is to facilitate the deparaffинization rinse. The probes, similar to LS probes, we now have VS probes 2.5, which contain this new buffer, but has the same probe design. With the amplification components, we're introducing our new RNAscope 2.5 amplification components, where all the similar changes apply. Where we've changed the components, there's no need to warm up no need to warm them up, and lastly, there is still no change to the amplification tree. The detection substrate remains the same. And lastly, the software is updated to version 4.0 to accommodate the RNAscope 2.5 assay. And please contact both ACD and Ventana sales to schedule an RNAscope VS 2.5 training and software update. Great. And now I'm going to introduce Courtney Edison, who will talk about RNAscope in the lab.
Thank you very much, Vinay. So I'm going to be discussing a study that we performed in the lab, which we've entitled The Sensitive and Specific Detection of Gene Expression at the Single Cell Level in Human Tissues using the RNAscope 2.5 product line. Now, as many of you are aware, detecting RNA biomarker expression at the single cell level while preserving spatial information is critical to understanding cellular organization and cell-to-cell interactions. Traditional RNA-ish methods are limited by sensitivity and specificity concerns. The data I'll be presenting here today will be highlighting our RNA-ish technology, the RNAscope 2.5 line, which can overcome the challenges of traditional RNA-ish while providing the single-cell expression resolution with spatial and morphological context. So some of the key highlights of the RNAscope 2.5 assay are we have a high signal-to-noise ratio, and this is determined by having intense punctate spots with little to no background. We also have strong product reproducibility, which we demonstrate by showing lot-to-lot consistency as well as run-to-run consistency. In addition, there's an ease of workflow with our protocol, in which there's one standard pretreatment condition for most tissues, provided our tissue fixation protocol is followed. And on the various platforms, there are several benefits in ease of workflow. On the manual and Leica platforms, we spend less time pre-warming reagents and also diluting the reagents. All are pretty much ready to use. On the VS platform, it is now fully automated, so there's no pretreatment that has to be done offline, including the deparaffинization and boiling steps. Everything can be done on the machine. And lastly, we present a robust assay in which virtually any tissue using our standard tissue fixation protocol can be studied, as well as virtually any gene target on virtually any platform, and that includes our manual and automated assays. So the data I'll be presenting in this study, I'd just like to disclaim that all of this has been based on an internal study where tissues were fixed per our recommendations and experiments were run here at ACD by our in-house scientists. So the first data that I'd like to highlight is the high signal-to-noise ratio. One of the fundamental challenges of traditional RNA-ish is that it can generate low signal and high background, and this can really compromise the interpretation and results of the experiment. Using the RNAscope 2.5 assay on the Leica Bond RX automated platform, we observed little to no background using a negative control probe known as DAP-B in a range of human tissues. And what I've highlighted here is breast, lymph nodes, skin, and stomach. Conversely, a positive control probe known as PBIB showed intense punctate spots in multiple tissues. And what I've shown here on this image is breast, lymph nodes, skin, and stomach. Similar results were also obtained using the Ventana Ultra platform and the automated platform, where we also saw, again, DAP-B staining showing no background and PBIB staining showing strong punctate spots. Similar results were also seen with the manual assay, which I do not show here. Now, a high signal-to-noise ratio with low background and strong signal really makes it easy for scientists to visualize, interpret, and quantify the data. And so what we've shown here is that the 2.5 assay can generate a very high signal-to-noise ratio in the tissues that we evaluated, which really enables identification of the true signal without interfering with background. Okay. So the next thing I'd like to highlight is the reproducibility and consistency of the RNAscope 2.5 product line. To first test the consistency of multiple lots of RNAscope reagents, we selected 3 lots on the LS platform using HeLa cell pellets. We tested with both the manual and automated assays and examined detection of DAP-B, our negative control probe, as well as a positive control probe, TBP. The data I show here is for the LS assay on the Leica Bond RX instrument, but similar results were obtained using the manual assay. So in this image here, comparing these 3 lots, we see that there's no background with DAP-B and very strong, consistent detection of TBP in all 3 lots of reagents. And as I mentioned, similar results were seen with the manual as well as Ventana assays. Okay, so we also quantified this to confirm that we truly are seeing no difference between the 3 lots. And we quantified this using a Halo imaging software, which determined the average spots per cell. And across the 3 lots, there was no statistically significant difference in the average spots per cell. So this really indicates the high reproducibility of multiple lots of the RNAscope 2.5 assay. So then we next wanted to see if the run-to-run consistency was the same between performing the experiment on 3 different days using the Leica Bond RX machine and also using the same lot of reagents. So again, we used HeLa cell pellets and the probes DAP-B and TBP with our automated RNAscope 2.5 LS assay. And as you can see here, we do have very reproducible results that was seen each day on the Leica Bond platform. No background was detected with DAP-B, and TBP staining was similar on all 3 days with the automated assay. Similar results were seen again with the Ventana Ultra and XT automated assays. Again, we quantified this the number of spots here to confirm that what we're seeing was truly accurate. When we quantified the average number of spots per cell, we can see that there's no statistically significant difference in the number of spots we're detecting from the 3 different days or 3 different experiments performed. So this really demonstrates that we have consistency between multiple runs of the RNAscope 2.5 assay. And I should also mention in data not shown here that within a run on the Leica automated assay, we performed 2 to 5 replicates of a HeLa cell pellet stained with TBP, and we show similar number and intensity of the dots. So this demonstrates we have intra-run consistency. Okay. And then lastly, we wanted to confirm if we have uniform staining across an entire section. So we probed HeLa cell pellets with our control probes, DAP-B or polar 2A. To determine if we do have a consistent uniform staining. And indeed, when we look at the polar 2A staining, we have a nice consistent staining across the entire HeLa cell pellet. And this finding was consistent not only on the Leica, the manual assay, but also on the Leica Bond RX, VS Ultra, and VS XT platforms. Okay. Okay, now I'd like to show some examples of multiple tissues being used on multiple platforms and with multiple targets. Okay. So with traditional RNA-ish, much time and reagent can be spent optimizing conditions for every tissue being examined before one even starts the actual experiment. So to address this concern, 41 human samples, which encompassed 8 types of tissues, were tested using our assay on both manual and automated platforms. Our tissue fixation protocol with standard pretreatment conditions for each platform was used. These 41 samples passed our RNA quality control standards, and I show a subset of those samples in these following figures. Okay. So what I show here is the manual assay and VS Brown assay. We demonstrated a little to no DAP-B background in cervix, breast, lymph node, lung, larynx, and skin tissue. Whereas in each of these tissues, we demonstrated punctate spots for the human probe TBIB. Okay. Similar results were also seen with stomach and soft tissues. Okay. So these results all suggest that one standard pretreatment condition coupled with our tissue fixation protocol will work on multiple different tissue types. We do want to caution that if the tissue fixation protocol is not known, then pretreatment optimization may have to be performed. Okay. We also wanted to see if whether or not we can use our assay to robustly detect multiple different specific targets for each of the tissues presented here. So to do that, we picked a panel of target genes that were specific for the tissues. What we show here on the manual and automated assays are that we have very strong signals using the HPV HR18 probe in cervix, FGFR3 in breast and larynx tissue, and the IG-kappa probe in the lymph node tissue. Okay. We also were able to detect low expression of the PD-L1 gene, which is a low expressing marker, in both lung and skin. Now, a unique feature of the RNAscope assay is its availability on 3 platforms, including manual, automated Leica Bond RX, and the automated Ventana Discovery Ultra and XT platforms. Showing the cervix tissue here probed with either DAP-B or Polar 2A. And what you can see is that in the cervix, we detected comparable Polar 2A staining across the platforms with no DAP-B background staining. I should mention that similar results were seen in skin and lung tissues as well. So for these particular tissue types and control targets, in our hands using our tissue fixation protocol, we observed equivalent staining across the manual and Leica and Ventana platforms. So overall, what we've shown here is that we have an RNAscope 2.5 assay, the new configuration of the current RNAscope technology. We demonstrate with this product high signal-to-noise ratio with little to no background. We also have a reproducible and consistent product. And it's a robust assay that can be used on virtually any tissue for any gene. And lastly, there's also an ease of workflow with our protocol. Less time is spent optimizing pretreatment conditions, as well as diluting and pre-warming the reagents. I should again reiterate that the optimizing of pretreatment conditions is provided tissue is prepared using our standard fixation protocol. And also the RNAscope 2.5 product line on the VS systems is now fully automated. And so with that, I would just like to pass this over back to Vinay. Thank you.
Thank you, Courtney, for highlighting the key aspects of RNAscope 2.5 that we've just launched this week. In summary, RNAscope 2.5 product platform covers our single-plex chromogenic and pretreatment reagents for manual and the Leica and Ventana automated systems. The product highlights improved robustness, ease of use, and improved performance. For more information, please go to our website at acdbio.com. On this website, you can get a quote and order directly for U.S. customers. For all other customers, please contact your sales rep. On the support section for acdbio.com, you'll see what's new with 2.5. There's also checklists and much more support information. So thank you for joining our RNAscope 2.5 webinar. We'll now pause for questions. So one question that's come in is, why should I switch to RNAscope 2.5? The new 2.5 assay will be easy to use and more reproducible, resulting in improved product performance. The manual chromogenic brown and red kits will require fewer steps for tissue optimization, and detection reagents no longer need to be pre-warmed. So one question that's come in is, would older probes from RNAscope 2.0 work with the new 2.5? For the manual chromogenic red and brown kits, the RNAscope 2.0 probes will work. For the LS and VS RNAscope 2.5, you will need 2.5 probes. So please do contact your sales rep for further information. One question that's come is, when will the RNAscope 2.5 VS red kits be available? These kits are readily available now. One question is elaborating on the standard fixation protocol. What we're seeing with RNAscope 2.5 is that many tissues now will follow our standard conditions. So we encourage you to please look at the manual where our standard conditions would apply for most tissues. Another question is elaborating on the steamer protocol. So the steamer protocol is essentially using an electric steamer that for RNAscope 2.5, we have validated our assay on the Oster steamer that could be purchased externally. This is essentially works as well as a food steamer in the sense that that the target retrieval solution is actually being steamed via the electric steamer. For more information about the steamer protocol, please consult our sample prep user guides that are on the web. And this sample prep user guides for 2.5. One question has come in is for the protease plus, can it be used instead of pretreat 2? The protease plus cannot be used in lieu of pretreat 2. Pretreat 2 is our target retrieval. Protease plus is our protease. Another question is protease plus, can it be used instead of pretreat 3 or 4? We recommend that you go to acdbio.com to consult the pretreatment matrix. Depending on your various tissues, this will require different proteases. One question is on the Bond RX, do we still need to use the detection kit from Leica for the new 2.5 red kit? The answer to this is yes. You will still need the Bond Refine Red kit to run RNAscope 2.5 RNAscope 2.5 Red. One question is, I'm in the middle of a project, can I still order RNAscope 2.0 kits? Yes, RNAscope 2.0 HD will be in stock for a limited time. We do encourage you to contact your sales representative for more information. Another question is, I still have leftover pretreat 1, pretreat 2, and pretreat 3. Can I use them with my 2.5 kits? The answer to this is no. RNAscope 2.5 HD has been optimized with RNAscope hydrogen peroxide, target retrieval, and protease plus, as well as protease 3 and protease 4. A similar question is, if I have 2.0 detection kits, can I use them with the new 2.5 pretreatment reagents? The answer to this is that for chromogenic red and brown, we specify that 2.5 pretreats should be used with the 2.5 detection kits. However, our 2.0 multiplex and 2.0 duplex can also use the new pretreatment reagents. Please consult the pretreatment matrix on the web. One question has come in about working with bone. Will 2.5 RNAscope allow me to work with decalcified sections? Unfortunately, we have not tested RNAscope 2.5 with bone, so we're unsure on the signal. One question is the cost comparable? As I mentioned earlier, the 2.5 kits remain the same price as the 2.0 kits. One question is, will 2.0 still be available for purchase, or will it be discontinued? So 2.0, so our 2.0 assay will still be available for purchase for those with special projects. We will be officially discontinuing RNAscope 2.0 by next year. It will be officially discontinued on June 1st, 2016. And while RNAscope 2.0 is not listed on the website, please do consult your sales rep. One question is, which pretreatment reagents should I use with RNAscope 2-plex or fluorescent multiplex detection in reagent kits? For optimal results, use the new pretreatment reagents, RNAscope hydrogen peroxide, target retrieval, protease plus 3 and 4, depending on your tissue types. One question is, are there any pretreatment recommendations as far as the new 2.5 versus 2.0? For this, to answer this question, this really does depend on the tissue types. So we do recommend that you start out with our standard pretreatment conditions. Thank you for joining the webinar. Since we don't see any further questions, we'll be ending this now. This webinar and recording will be up on our website by the end of next week. Thank you very much for joining.